Welcome back to Acing AP Biology. In this video, we will be discussing the plasma membrane. The structure of the plasma membrane is described using the fluid mosaic model. The plasma membrane is fluid because the phospholipids can move laterally due to weak hydrophobic interactions like London forces that hold the tails together. However, they do not move transversely because the polar phosphate heads would not cross through the nonpolar tails. The proteins embedded in the membrane move laterally as well, but they move more slowly than the phospholipids. Membranes must be fluid to function efficiently. The plasma membrane is also known as a mosaic because it exists as a mosaic of lipids, proteins, and carbohydrates. The plasma membrane has several major functions: protection of the cell, regulating the interactions between the outside and inside of the cell via its selective permeability and attachment to the extracellular matrix, which is the network of specialized proteins and carbohydrates outside the cell. Transport, cell signaling, cell-to-cell recognition, enzyme activity, and the joining of neighboring cells, known as intercellular joining. Now let's look over a diagram of the plasma membrane. As you can see, it is a mosaic of different macromolecules present throughout the phospholipid bilayer. Notice that some fatty acid tails are angled. This is because it is an unsaturated fatty acid tail, and so it forms a kink. The kinks aid in spacing out the membrane, preventing too much rigidity, and thus helping maintain membrane fluidity. Cholesterol, present in the membranes of eukaryotic cells, have the same purpose. In cold temperatures, eukaryotic organisms regulate membrane fluidity by increasing cholesterol and unsaturated fatty acid tails in the bilayer. In warm temperatures, cells must ensure that the membrane does not become too fluid, so it decreases the amount of cholesterol and makes more saturated fatty acid tails. There are two different locations for proteins in the plasma membrane. They can be integral proteins, known as integrins, that are transmembrane proteins, meaning that they span across the entire membrane. These proteins are amphipathic because they have hydrophilic regions near the ends and hydrophobic regions in the middle. They are mainly involved with transport, enzyme activation, and cell signaling. Peripheral proteins exist on the surfaces of the plasma membrane. They can be attached to integral proteins, anchored to the extracellular matrix, or on the inside of the cell, be anchored by the cytoskeleton, specifically microfilaments. Carbohydrates are attached to the outside of the cell. They usually exist as oligosaccharides, either bound to a protein, forming a glycoprotein, or bound to a lipid, forming a glycolipid. Both of these aid in cell-to-cell recognition, serving as ID badges for the cell, helping the body differentiate between its own cells and foreign material. This is key for the organism's immune response. Proteoglycans are composed of carbohydrates and protein and are an important part of the extracellular matrix. Collagen fibers provide support and strength to the cell as a major component of the extracellular matrix. Fibronectins attach the plasma membrane to the extracellular matrix by binding to integrins. Interactions between the inside and outside of the cell are mediated by fibronectins, integrins, and microfilaments inside the cell via signaling processes. If they give you a diagram of the plasma membrane on the AP, in order to figure out which side faces the outside of the cell, look for glycoproteins and glycolipids because they only exist on the outside of the phospholipid bilayer. That's it for this video. In the next lesson, we will discuss the plasma membrane's involvement with transport in and out of the cell. See you then. For more bio content, hit subscribe.